What I love is the unpredictability of what we do. So if you pursue interesting problems in an open-minded fashion, it's inevitable over time that you make discoveries and unexpected observations. And in my research career, some of these have actually turned out to be very important and useful. My main interest surrounds the molecule DNA, which of course is fundamental to all living systems. And we're interested in how the shape, structure, and chemistry of DNA gives it its properties. Our research is by nature fundamental. We explore new things, undiscovered phenomena, and along the way we tend to innovate and invent methods to study DNA. And some of these have turned out to be quite useful. So one that goes back 20 years actually, in a collaboration here, we invented a new way of decoding the sequence of DNA. This eventually led to a biotechnology company, and today this is the method used globally to sequence human genomes and the genomes of other organisms. More recently, in one of our studies, again we invented a technology for detecting features called epigenetic features in DNA. And this too turned out to be useful and has been commercialized and is used quite broadly around the world. There are two main areas we work on, both on DNA. The first concerns the structure of DNA. Now everybody assumes that DNA is the famous double helix that Watson and Crick discovered and published in 1953. We have actually been studying a four-stranded DNA structure called the DNA G quadruplex, and we have shown that this structure forms in human cells, and we've been trying to figure out what it's doing and its impact on biological function. And very recently, in fact, we've published some work showing its importance specifically in certain cancers and its potential as a cancer drug target. The second theme is to do with the chemistry of DNA. Now, DNA has four letters, G, C, T, and A, and they constitute the genetic code. This has been known for some time. However, what's quite new is the discovery that some of these letters can undergo chemical changes that occur naturally in living systems. And so, for example, the C letter actually has at least 4 different derivatives. So what we've been doing is studying this dynamic chemistry that occurs naturally in DNA and trying to understand what its function is. My family actually emigrated here from India, and I was born in India, and so I was certainly the first person to go to university in the UK from my family. I grew up in the north of England and went to a comprehensive school, and so coming to Cambridge from that environment was definitely a culture shock for me. Quite different, in some ways quite intimidating compared to the environment that I came from. However, what I would say is over the years as a student, and then eventually some years later I travelled and came back as an academic, Cambridge is now my home, and I'm very comfortable here. We're quite a large group, we're about 30 people. We actually have 2 laboratories, one in chemistry and another one at the Cancer Research UK Institute, but we work as a single team. Innovation and invention is really at the heart of what drives scientific progress, so we have large and small interactive sessions where we brainstorm ideas. So these can be very open and free-flowing activities. It can be a lot of fun, and we don't always do it in the department. Sometimes we do it outside, sometimes we do it down the pub. I've been a big fan of having group retreats. We've been to the Alps, we've been to Tuscany, we've also been to interesting old buildings around the UK. And then we have a mixture of scientific discussion activities as well as sort of more social activities, just to make sure that the communication is flowing and we all get to know each other, which is important because we work as a team. very important to me that everyone enjoys what they do. I love what I do, and I want all my group members to enjoy it as much as I do.